right, welcome everyone to the front row. My name is Jamie Williamson. I'm a professor here at the Scripps Research Institute, and I'll be your host this afternoon. I understand it just rained a little bit. That's kind of unusual, but it's not a forecast of doom and gloom for the presentation, I assure you. It's going to be sunny and rainbows. So let me welcome everyone. We've got about 250 people. We've got a good solid crowd in person. We have about 3,000 that are attending virtually, so welcome to all of you. Now I've really messed things up. Okay, so today our speaker is Ben Cravatt, and I thought I'd give a little bit of an introduction. So in 1964, Arthur C. Clarke wrote a book, and one of the real famous passages from this book was that any sufficiently advanced technology is indistinguishable from magic. And he was one of these visionary people that really tried to project about what is possible and going beyond what is possible to the potentially impossible. And so I would say that Ben is, in fact, someone who has executed this in his career. And so I would add a corollary to this, and that is anybody who invents technology that is indistinguishable from magic is, in fact, a wizard. And so Ben is some kind of scientific wizard. So let me see if I can explain, in a very crude analogy, and he will eloquently, tell you what he's doing. But imagine you do an experiment. Your grandkid comes over to your house, and you say, "Hey, let's play ball. Do you want a baseball, a basketball, a football, or a soccer ball?" And they choose basketball, and off they go, and they're shooting hoops. You've just done an experiment, and you learned something. That kid likes to play basketball. So now let's think, we're going to do this on a massive scale. So you go into a gym, and you just fill the gym with every imaginable kind of ball or sports equipment. And then you let the entire school into the gymnasium, and they run around and they pick out the thing that they're most interested in, and suddenly they're all playing with balls. So you've massively parallelized this. And if you look at the gym, you have just gotten a lot of information about what the kids are interested in. So Ben's wizardry is doing this, except he does this in a cell. So the cell is the gym, and it's not balls, it's molecules. So he basically takes all kinds of compounds and mixes them up with enzymes and learns about what the enzymes are doing in the cell in this massively parallel format. And this is, I submit, indistinguishable from wizardry. It's really a remarkable platform. And actually, and we'll discuss in a sec, but this is the kind of place where this platform can be developed. We have incredible colleagues. They're amazing chemists and amazing biologists. And this is it makes a cauldron of innovation. This is where you can do this kind of science. And it serves as the basis for the next generation of drug discovery, because many, many drug targets are in fact enzymes. And Ben's technology allows you to accelerate the pace of drug discovery. And so, it's no accident that, I mentioned Arthur C. Clarke. He wrote 2001: A Space Odyssey, which was a futuristic vision of the universe. And so Ben's role at Scripps and in the world is to expand our understanding of how to make drugs. So I can say a few things about Ben. He's probably going to give a little self-introduction, but Ben was a graduate student here, and we were fortunate enough to retain him. He's been here essentially his entire career since he was a PhD. He's won a lot of awards. I'll just single out the Wolf Prize, which is just one of the most prestigious science prizes in the world. And he's already received that, and let's just say we're optimistic about additional accolades. So I'm going to now invite Ben up to the stage. So at the end, I'll come back and we'll have a little Q&A. So if you're in the virtual audience, you can type in at any time a question. There's a lot of you, so I can only get to some of them. But I'll have an iPad. It can be relayed to me. If you're in the studio audience, there will be microphone runners. Put your hand up, and they will come to you, and we'll have a little discussion at the end to reflect on Ben's presentation. So let's welcome Ben Cravatt.
Thanks, Jamie, for that kind introduction. Go Padres. Okay. Okay, so thank you all for attending this afternoon. I'm excited to share with you our lab's work and efforts to try to expand the drug target universe. As Jamie mentioned, I thought I'd give you first an introduction to my background. I grew up in Los Angeles, and I always had a strong interest in science, but when I migrated north to perform my undergraduate work at Stanford, I was under the premise that I would become a physician. And admittedly, where I grew up in Los Angeles was really far away from any sort of university, so I didn't really understand what university professors did. I assumed they taught and maybe wrote a textbook or two and then called it a day, right? And then I started meandering around the Stanford campus and realized that the chemistry and biology professors actually mostly did research, and I had one of these wait what moments where I was like, wait, what? You can actually get paid to and make a living off of discovering how life works and developing ways to treat its disorders? And I was like, sign me up, right? So that was pretty much the moment that my career direction changed, and I was really fortunate to work in a chemistry lab at Stanford while I studied biology, and I got super interested at the interface of those two fields. And so when I pursued graduate education, that's where I wanted to be. At the time, when I entered graduate school in 1992, there were all the major universities across the country. None of them had any programs that professed an emphasis on interdisciplinary science between chemistry and biology. In fact, there was only one program in the entire world that had that as a mission, and that's right here at Scripps Research, okay? And I had the good fortune of working with the then president of Scripps, Richard Lerner, and Dale Boger, who's in the audience today, as my mentors, and Richard and Dale and individuals like Bernie Galula pioneered the training and education of students at the interface of chemistry and biology. And I say that to you because if you visit any university in the world today, not just here in China and Europe, every program has an emphasis on chemical biology. But that all started where you're sitting today at Scripps. And so I'll have a few more words to say about this amazing institute at the end. As you can see, I've been here ever since. I'll never leave this place. So, okay, so if there's one message I will hope you will take away from this talk, it is that we are living in an incredible time for biomedical research. We are the first generation of scientists who can select problems to study in the lab based on deep knowledge of human biology and disease. And we're going to spend a bit of time throughout the talk today why that's so important for accelerating not only disease understanding, but ultimately the development of medicines. But to give you a bit of a context for what I mean by deep human biology knowledge, this is one of my favorite databases that's publicly available. It's called the OMIM database. This database houses a collection of all known monogenic human disorders related to their causative genetic basis. And if you look at when I was starting graduate school, this wasn't much of a database, right? There wasn't much information at all on this. And that's because it took labs 30 or 40 human years to map the causative genetic basis of a single disease. That's actually how cystic fibrosis was deduced to be caused by CFTR mutations. Incredibly laborious process. But as you can see, as I traverse through graduate school, this database starts to get populated a bit, right? And so what was happening during this time? There were advances in DNA sequencing technologies which allowed the communities to begin to sequence genomes on a global scale, right? So in between, when I was starting and finishing graduate school, the yeast genome was sequenced, and shortly after, the human genome was sequenced. And then you see this incredible acceleration to the point now where this database essentially houses every known monogenic disorder with a definition of its underlying genetic cause. For those of you that are lifelong San Diegans, companies like Illumina have contributed significantly to the technologies that have allowed this type of accelerated understanding of gene-disease relationships. In parallel with those efforts, we've also seen advances in gene editing technologies, technologies that allow us to manipulate any gene in any biological system. You'll know these in the terms such as CRISPR technologies. These also have created incredible databases for chemists and biologists to interrogate, to understand, and select protein pathways to not only study, but to pursue for drug development. One of my favorites is what's referred to as the cancer dependency map. This is a database created by the Broad Institute, where they essentially went in and genetically knocked out every gene in thousands of cancer cell lines and asked which cancer cell lines depend on which genes for growth. And what's remarkable about that database is it can rediscover essentially all validated intrinsic cancer drug targets for which we have medicines today, including the estrogen receptor in breast cancer, the androgen receptor in prostate cancer, KRAS and EGFR for lung cancer. But what should excite you even more is when you look at where those proteins reside on this graph, there's almost 1,000 other gene products that have similar dependency relationships in cancer for which we have no medicines yet, right? So that's the opportunity and the hope in the future that this type of untapped relationship, molecular relationship between disease and genotype can be exploited. So you might ask, why haven't we exploited those relationships yet? And this gets into the nuts and bolts of today's lecture. So I think over the last 50 plus years or so, we've gotten really good at creating drugs for specific types of proteins that are deregulated in disease. I would call these here today classical druggable proteins. They have a really nice small molecule binding pocket, largely because that's what they naturally do. They normally bind small molecules. The androgen receptor or the estrogen receptor binds hormones. Kinases like EGFR bind ATP, and chemists have gotten really good at creating highly potent and selective ligands that bind those pockets that can become drugs. The challenge is that all these new drug targets that we are illuminating through methods like CRISPR and genome sequencing, many of them look like this. They're flat and they have no deep binding pocket for creating small molecules to exploit. In fact, our classical approaches for trying to drug these proteins end up resulting in an outcome that looks like this. And the small molecule just goes away, okay? So this is the challenge that our lab and many groups at Scripps face that are interested in drug development in a future-looking manner. How do we create drugs for proteins that lack deep pockets and instead possess cryptic, dynamic, shallow pockets? And we're going to discuss how our lab has developed technologies today to try to address that challenge. So those types of problems have led to these sorts of graphs that you can find in review articles in our field, where there's a portion of protein space in our cells that harbors these classical druggable proteins, right? These ones with deep pockets that I mentioned earlier, and that's a static portion. We know those proteins pretty much completely, and we've largely exploited them and tapped them out for drug discovery opportunities. And then there's this ever-expanding portion of our cells that contain proteins that have strong human biology disease relevance that we haven't yet figured out how to drug. And our lab and others are trying to develop methods to address that challenge. To summarize this brief intro, I think this is how I encapsulate it. If you're ill and you go to the doctor today, it is much more likely today than it was 20 years earlier that the doctor will be able to tell you mechanistically what is wrong with you. You will have a diagnosis that has a molecular definition of what's causing your disease. But to be frank, I don't think we've gotten much better at treating those diseases, right? And that's, of course, of limited value to the patient to know what's wrong with them and be told we have nothing to do about it, right? So our aspiration is to somehow enable disease treatment to catch up and match disease diagnosis. And I'm going to introduce you to a technology today that we refer to as activity-based proteomics as one way to address that challenge by bringing, as Jamie alluded to, candidate drug molecules into living systems and interpreting what proteins they bind to. I'm then going to explain how that technology has enabled us and the industry to resurrect or restore interest in a aged modality for drug development that is actually the source of some of the most famous drugs that we're aware of, but yet for many years was shunned by the pharmaceutical industry as an approach called covalent chemistry, or as I'll refer to after here in the talk, permanent chemistry, okay? So I'll introduce you to this method, and then we're going to jump into how it has enabled permanent chemistry to become a modality for drug development that is expanding radically the target universe. Okay, so let's first start with how are drugs normally discovered, at least small molecule drugs. Here's a human cell. It harbors a bunch of proteins that have different types of pockets that might bind small molecules. If a drug pharmaceutical company is interested in one of these proteins, the first thing they'll do is usually take it out of the cell, put it in a test tube, and then develop an assay to screen for it to bind small molecules as candidate drugs, okay? This has been a tried and true approach for drug development. It has worked over and over again throughout the years, but it has some serious limitations. As Jamie already mentioned, this is your kid with one ball type of problem, right? This is a one protein at a time type approach, very laborious to commonly express and purify proteins, develop specific assays for them, a process that we'd like to accelerate. Two, you've taken the protein out of its native environment, right? So to use Jamie's analogy, this might be like handing a kid a basketball and putting him on a soccer field, right? There's only so much a kid can do with a basketball on a soccer field, right? These proteins exist in cells in different modification states, binding different partners, different neighbors, in different localizations, and all that information is lost when you put them in a test tube, and that information can actually impact how drugs work, right? So the last thing you want to do is spend years developing a drug that works beautifully against a protein in a test tube. You put it back in the cell, and it doesn't work anymore because the protein's in a different state, okay? So what the activity-based proteomics technology allows us to do is to bring chemistry into the cell, okay? So instead of taking the protein out of the cell and asking how it interacts with chemistry, we're going to bring chemistry into the cell and develop a method that tells us how those compounds bind proteins directly in their native settings, right? So if one of these compounds binds a protein in the cell, we've developed these broad reactive probes that can tell us that event has occurred, record it quantitatively, and read it out in a quasi-universal manner, right? So this method allows you to now look at thousands of proteins in parallel. That's where the term proteomics comes from, right? That means a omics scale analysis of proteins, looking at many proteins at once. And what's perhaps as important for this methodology is that it reads out how compounds bind proteins in their endogenous natural setting. So if a protein's modified, or if it's bound to another protein, or if it's localized somewhere specifically in the cell, we can read out how compounds bind those proteins in those settings, right? So we're much more likely to find small molecules that will retain activity as drugs in the disease-relevant states in which they operate, because that's where we discovered the drug candidates in the first place. Okay, so with that background, let's move on to the integration of activity-based proteomics with permanent chemistry. This begins with a story dating back about 20 to 25 years that began in our lab with an interest in a signaling pathway in our brains called the endocannabinoid pathway. As the name endocannabinoid might imply, there are exocannabinoids, the most famous of which is this compound shown here. This is tetrahydrocannabinol, or THC, the psychoactive ingredient in cannabis, okay? So when THC enters the brain, it produces virtually all of its neurobehavioral effects through a single protein, a G protein coupled receptor called cannabinoid receptor 1, or CB1. And we've known for millennia that some of these effects are medicinally beneficial, including reductions in pain sensitivity, improvements in anxiety, but these also come with a variety of untoward side effects, including motor defects, cognitive impairments that have limited the broad utility of direct cannabinoid agonists as medicines. This system, of course, did not evolve to recognize THC. There are endogenous ligands for the cannabinoid receptor, which are shown here, these lipids, referred to here as anandamide and 2-arachidonyl glycerol. They bind the cannabinoid receptor with actually pretty similar potency and affinity as THC. But unlike THC, if you administer these compounds to an animal, they don't show any evidence of experiencing cannabinoid-like behavioral effects. And that's because they're rapidly metabolized by enzymes, okay? And enzymes inactivate these compounds. And that led to the hypothesis in our lab and in the pharmaceutical industry that if you can create compounds that could target those enzymes, you might be able to elevate these endogenous signaling molecules, let them signal more loudly where they normally signal in the brain, and produce an interesting subset of the behavioral effects you see with direct receptor agonists like THC or cannabis. That's a classical approach, by the way, for neurobiological drug development. Drugs like Prozac operate by binding a channel that elevates serotonin levels, and then serotonin goes on and produces more activity through its receptor. Acetylcholinesterase inhibitors that are used to treat dementia and Alzheimer's bind to the acetylcholinesterase enzyme, elevate its endogenous neurotransmitter acetylcholine, again, creating more signaling. So our lab actually played a pretty strong role in identifying enzymes that regulate endogenous cannabinoid signaling, including this fatty acid amide hydrolase enzyme shown here. So this enzyme will hydrolyze this amide bond and terminate the activity of this endogenous ligand for the cannabinoid receptor. It actually is an interesting enzyme. Here's the structure we solved with Ray Stevens' lab here at Scripps. It's a membrane-bound enzyme. It probably extracts these lipids directly out of the bilayer to perform its chemistry on them. And so we had the great fortune to work with Pfizer to create some of the first clinical candidate drugs for the THAW enzyme. The idea here is these compounds would, again, block this enzyme and elevate endogenous cannabinoid signaling to produce useful medicinal properties. And so at this point in time, this is around 2005 timeframe, Pfizer ran a traditional pull the protein out of the cell, stick it in a test tube, screen. And they got some really potent inhibitors of FAW. And this program was moving along in a seemingly swimmingly manner. And then one day I got a call from our collaborator at Pfizer, Kay On, and she said, we have a big problem. And I said, what's the problem? And she said, well, we've been doing mechanistic studies on these compounds, and we're pretty sure they operate by a covalent, irreversible mechanism. And with Ray Stevens' lab, we went on to show that indeed, when these compounds bind FAW, they form this permanent, what's referred to as a covalent bond with the enzyme. Okay? It actually sounds kind of neat, right? You've bound the protein permanently, it's totally shut down. So why is Pfizer concerned about this? Well, at that point in time, 2005, 2007 timeframe, the pharmaceutical industry was exclusively interested in developing compounds that bind proteins reversibly, okay? They'll bind tightly, but they'll be able to fall off once the protein's denatured and disassociate. This is how drugs like Lipitor and Januvia work. They target deep pockets on proteins, they bind those pockets reversibly and fall off to treat diseases like cardiovascular disease and diabetes. Now, having said that, however, some of the drugs that you guys are most familiar with actually operate by an irreversible permanent mechanism. They bind proteins, and then they form a physical bond, and they don't let go of the protein. That's how drugs like penicillin and aspirin work, okay? So you might ask, if penicillin and aspirin work that way, why is the pharmaceutical industry so concerned about this type of drug mechanism, this permanent drug mechanism, for their own programs? Well, first, you have to realize that these drugs were not discovered to be permanent drugs intentionally. These were discovered by phenotypic screening, and only actually after they were already approved, in some cases, was it mechanistically understood that they operate by this permanent mechanism. So I would say these were serendipitous discoveries that were begrudgingly tolerated by the pharmaceutical industry. And so their concern is the following. If these drugs can permanently bind to your protein of interest, maybe they can permanently bind to hundreds of proteins in our bodies to create lethal toxicity, okay? And this admittedly was, in my view, largely conjecture in the early 2000s, but it was entrenched in the pharmaceutical industry. In fact, our lab was using activity-based proteomics to discover covalent ligands and inhibitors for enzymes during this time, and I would give talks at meetings, and almost without exception, senior representative from a pharmaceutical company would come up to me at that time after the talk, and they said, that was a cute talk. But just so you're fully aware, no one's ever going to do this in the drug discovery industry. There will never be an intentional effort to develop permanent covalent drugs for proteins. You're nuts. And I said, well, thank you for that input and that feedback. And I wasn't dismissive of it. It's not an invalid concern. I just felt like if we could generate data that could challenge that notion that these drugs would be this promiscuous and non-selective, maybe we could change some minds and open up an opportunity for this type of permanent modality, as we'll discuss in a few moments, to drug proteins that have been historically undruggable. And fortunately, with this program with Pfizer, they were very forward-thinking, and they said, show us data that says that these permanent inhibitors of the Fa enzyme are selective. And this is one of the first instances where the activity-based proteomic technology had a fundamental impact on drug development, because we could take that Pfizer compound, we could add it to mice, sacrifice the mice, take their brains, run an activity-based proteomic experiment. Every one of these bands on this gel is a protein that in principle could react with this compound, and only one does. And that target is the Fa enzyme, the exact target to which the drug was developed, okay? So this type of target specificity was what began to de-risk the notion that these sorts of permanent chemistries are going to be promiscuous in the body. In fact, we could even take this one step further, because around this exact same time, Barry Sharpless at Scripps invented click chemistry, which he was awarded the Nobel Prize in 2022, which allows us to even convert these compounds themselves into activity-based probes by appending a simple little alkyne group that allows you to click on a fluorescent tag and look directly at what this compound reacts with across an entire tissue or cell. So when you add this compound to mice, you take the brains and say across all proteins that exist in the mouse brain, what reacts with this compound? You see it reacts with one protein, the Fa enzyme, and nothing else, its intended target, right? So it was this type of single target specificity across an entire protein content of a tissue like the brain that began to convince the pharmaceutical industry that permanent chemistry might be more selective than they initially anticipated. And now there are several Fa endocannabinoid inhibitors in clinical development. I just wanted to highlight one for you here that I'm particularly proud of, because the concept originated from our lab back in the 2005 to 2009 timeframe. This was work done by Jonathan Long, who was a student here at Scripps and is now a professor at Stanford. John created compounds that bind multiple enzymes in the endogenous cannabinoid system, a single compound that could bind multiple targets in an intentional way. These compounds then went into a biotechnology company that we co-founded with Dale Boger, who is here today, Abide Therapeutics. They advanced those compounds into clinical candidates, and Abide is actually still here today as a innovation arm of Lundbeck Therapeutics. And those specific compounds are now with Bristol Myers Squibb, where they are in phase 2 clinical development studies to treat multiple sclerosis, in particular spasticity in multiple sclerosis, and agitation in Alzheimer's, two major unmet needs in neuroscience, okay? If you go back to this slide, however, you'll notice this is a long process, right? Almost 20 years from initial discovery in an academic lab to a phase 2 proof of concept study. And we don't want to have to wait that long, right, for discoveries to go from the academic lab into a clinical investigation. And we're going to return to how deep human biology knowledge is allowing us to accelerate that process, right? Because remember, back in 2005, we didn't have the knowledge we have today, right? It's only in the last 20 years that this type of knowledge has been accumulated. And I'm going to give you one example of how fast this process can now go based on human biology information. Okay, so if you were to ask why our lab, Pfizer, Abide, and others have been so successful at creating high-quality permanent chemistry for enzymes like the Fa enzyme, the honest answer is we kind of cheat. And we cheat because certain proteins in our bodies possess such reactive residues in their small molecule binding pockets that it gives the medicinal chemist a tremendous sandbox to come up with complementary chemistries that will react with that hyper-reactive residue and leave all other proteins unperturbed. That sounds great, and it actually is great, but it only applies to about 1% of all proteins in our bodies, right? So that's a narrowing funnel of opportunity. As we consider ways to extend permanent chemistry beyond a hyper-reactive residue in the active site of an enzyme, there are other residues on our proteins that are intrinsically reactive, such as a residue referred to here as cysteine. And these residues, in principle, could provide a way to create permanent chemistry against any and all proteins that exist in our bodies. And in fact, shortly after our work with Pfizer on the FA endocannabinoid metabolizing enzyme, these sorts of compounds began to emerge in the pharmaceutical industry. This is a drug sold today under the name Imbruvica. It's called ibrutinib. It is a compound that binds a kinase active site called BTK through permanent chemistry using this group down here. This is a pretty famous drug. If you're into reading books about biotech, it's the focus of this book, For Blood and Money, which actually has some really good science in it. It's mostly like biotech soap opera stuff, right? So, but if you're into that, it's a good read. But to Vardy's credit, the author, he spends a decent amount of time in this book on the exact topic I'm sharing with you today, that this compound was taboo at that time. Like, it was created by Celera, a small biotech company that actually originally started as a genomics company, by this chemist, Zhengying Pan, okay? And no one took it seriously at that time as a drug candidate because it operated by covalent chemistry, right? Permanent chemistry. And the industry said, this is not the way we develop drugs. But Pan's a smart chemist, right? So if you look back at the first paper that discloses this compound's activity, in figure 1, you will see not only the structure of what became the drug, but the structure of this compound shown here, the drug attached to a fluorescent tag. This is basically an activity-based probe, the type of technology I've already introduced you to, which allowed Pan to take this compound, add it into cancer cells, and show that across all proteins in those cancer cells, only the intended target, BTK, reacts, and no others, right? So again, these types of innovations in chemical technologies, like activity-based proteomics, de-risked the concept that these molecules could be highly selective for their intended targets. And believe it or not, now today, there are almost a dozen cancer drugs on the market that operate by permanent chemistry, targeting cysteine residues in the active sites of kinases. I show some of them here too. Some of these are multi-billion-dollar drugs, not just ibrutinib, but osimertinib or Tagrisso for lung cancer. Actually, every pharmaceutical company now intentionally practices covalent chemistry or permanent chemistry. Now, if we said all that, I would still say that these efforts have their own unique cheat code, okay? And their cheat code is each of these kinase targets that they're going after has a very deep small molecule binding pocket, okay? So in almost all of these cases, the first compounds that were discovered were classic reversible ligands, okay? And then later on, they were retrofitted with chemistries to make them permanent. So in other words, they start out like this, right? And then they end up like this, okay? And that's great. There's nothing wrong with that approach, a fantastic approach, right? But I'll remind you that modern human genetics tells us most of the targets that we want to drug today that we can't yet drug look like this, right? So you can try to develop a reversible molecule, and it's just gonna bounce off, right? So we have to come up with another way to unlock this portion of protein space for drug development. And this began an effort in our lab that dates back 10 or 15 years and is still the major focus of what we work on today. And that was to intentionally develop small molecule libraries from the beginning that operate by permanent chemistry and ask whether, using activity-based proteomics, we can use this type of chemistry to discover compounds that bind weakly to proteins initially reversibly and then lock on through a permanent mechanism to allow us to drug shallow, dynamic, cryptic pockets on proteins that would have been resistant to traditional drug development methods. This is probably the most science-intensive slide, and you don't really need to learn anything about it other than to know that this method for discovering permanent chemistries that bind proteins has no cheat codes, okay? It can allow us to discover any small molecule that binds permanently to any protein in any biological system, okay? And we leverage Modern technologies like mass spectrometry to achieve this objective. These are technologies that were pioneered by John Yates's lab here at Scripps, for instance, to allow us to look at directly the sites on proteins that bind permanently to these reactive compounds. And when members of our lab, like Carrie Bacchus and others, began to apply this technology globally, we began to get our first glimpses into the power of permanent chemistry to radically expand druggable space in our cells. She identified hundreds and hundreds of proteins that could be targeted by permanent chemistry. And when you inventoried those proteins and matched them to databases that harbor known drug targets, what you found was the vast majority of them, this large white slice of the pie, this was the first evidence for those proteins interacting with small molecules. So these were all previously undruggable proteins that now were being unlocked by permanent chemistry read out in the cell with activity-based proteomics, coming from all types of protein classes, right? So this taught us that permanent chemistry can allow us to target shallow, cryptic, dynamic, druggable pockets on proteins that were resistant to methods used previously. At that moment in time, we launched another biotechnology company. This was with my colleagues here, Phil Barron and Jin Kuan Yu. I see Jin there. Vividion Therapeutics. And this involved Tom Daniel and Rich Heyman. Tom's also in the audience as mentors and co-leaders in this company's development. I want to say that one of the incredible things about the San Diego community is we have outstanding biotechnology leaders like Tom and Rich that spend most of their day paying it forward, giving back to younger generations, helping them learn how to build companies, to traverse all the challenges associated with those efforts. And I'm forever indebted to individuals like Tom and Rich for the guidance they've given folks like Jin, Phil, and me as we try to translate our technologies into companies that can ultimately build medicines that can benefit you, your families, and your loved ones. Vividion proceeded to industrialize that platform at a scale that I never could have imagined possible, allowing them to look at thousands of compounds for interacting with thousands of proteins in a week through permanent chemistry. I mention that because as someone who's launched a few companies out of our lab, I often get asked, when's the right time to exit technologies from your lab and put them in a company? And to be honest, that's one of the hardest questions that we have as academic scientists to answer, and there's really no perfect response. Each case has to be handled individually. But I would say that if it's too early, that means the company's doing academic experiments, and you don't want that, right? But on the other hand, something like this platform could have never been developed in an academic lab, right? So we often decide it's time to launch a company when we've done all we can do for a platform, and its next phase of development can only be done in a for-profit company setting. Vividion's now used that technology to bring multiple drug candidates into the clinic. I'm just going to highlight one for you here that targets this interesting enzyme called Werner helicase by permanently binding a cysteine at this dynamic pocket. In fact, when this drug binds, you will see the protein makes a whole variety of motions. So Dorothea Kern here is in the lab, and she's a new colleague of ours here at Scripps who is an expert in studying how small molecules affect and cause protein motional changes like this. I highlighted this example because I mentioned to you earlier that I was going to return to the notion of how we can leverage deep human biology knowledge to accelerate the discovery of medicines and bringing them to patients. And Werner helicase is a fascinating story. It actually emerged as a drug target from the cancer dependency map that I mentioned at the outset of the talk back in 2019. It was discovered that when this helicase is deleted from a specific type of cancer cell that has a genomic aberration called high microsatellite instability, which means that it repeats in its genome that most normal cells don't have, those cells can't grow if they don't have Werner helicase. The rest of our cells don't really care, okay? But these weird cancer cells that have these amplified microsatellites depend on this helicase for growth. That's called a synthetic lethality relationship in scientific terms. So that was in 2019, okay? Within 5 years, Vividion and a few other companies had developed clinical candidates to target this helicase with high specificity, and within 1 year, we have clinical data, right? So this timeline that I showed you previously of 20 years for the endocannabinoid hydrolase drug candidates has now been compressed to 5 years. And Vividion and Roche recently reported some of these data at a recent AACR meeting where you see substantial disease control rate and many clinical responses in patients being treated with these drugs. And these are patients that have failed all prior therapies, right? These are colon cancer patients that have gone through the types of immuno-oncology therapies that Travis Young, who gave the outstanding previous front row lecture, described. Failing all these therapies with nothing else available to them, they are now under disease control with this Werner helicase inhibitor. If you're interested in learning more about the compound, Timothy Yap, who's a clinical investigator at MD Anderson and leading the trials, has a couple of YouTube videos describing the impact of this drug on his patients and his excitement for this mechanism going forward. So you can consult that information publicly. Okay, so to wrap up on some future challenges and then a few words about Scripps. I've mentioned that the deep human biology knowledge that we're accumulating today has radically accelerated our understanding of the mechanistic basis of disease. And humbly speaking, there are still many diseases that we understand mechanistically that we can't treat. And I am, and will always be, a firm believer that innovations in chemistry will continue to break that problem down into digestible, solvable units. And I want to highlight some of the chemistries from my colleagues at Vividion, the Barron Lab, and the U Lab that continue to innovate on creating new types of chemistries that can allow us to drug shallow, difficult pockets that have not bent the knee yet to the historical chemistries that exist in the drug discovery industry. I also think that these methodologies and genomic methodologies are illuminating new relationships that will lead to new drug targets once we can understand mechanistically what is going on. I highlight, again, the cancer dependency map data shown here. These are a bunch of genes that all show provocative relationships where they are required for the growth of a distinct subset of cancer cells. In fact, the Werner helicase that I just mentioned to you is embedded in this group. But of these targets, or theoretical targets, most of them aren't being worked on by the pharmaceutical industry. So why is everyone huddling around Werner helicase and ignoring all these other ones? It's because Werner helicase, we know which cancer patients to treat. We know, because we can sequence their genomes, that those cancer patients have microsatellite instability, so we can select those patients. And this is one of the major challenges we face in cancer research. Almost all of these other relationships are probably going to be beyond the genome. They're going to be relationships that show dependency on specific signaling pathways in the cancer cells, signaling pathways that are not easily extracted from genome sequence alone. But what's encouraging for us is if you look at only our own labs' chemical biology efforts, we've already discovered ligands for a significant fraction of these proteins. So now we can use chemistry-first approaches to understand why certain cancers depend on these proteins, and then that will give us the patient selection strategy to carry them forward into the clinic. And then everyone will huddle around them, right? That's the goal, right? To make enough of an advance that every company wants to drug something, and then you have a whole variety of productive competition which benefits all patients going forward. Okay, so one final comment on a topic that I'm sure many of you are interested in, which is artificial intelligence and machine learning. As excited as we are about the methods in our lab for revealing cryptic druggable pockets through permanent chemistry, they won't find all pockets, right? Because some pockets won't have residues nearby that will allow us to attach permanent chemistry. And we've begun to wonder whether our deep maps of pockets that are enabled by permanent chemistry might be useful to predict other pockets that exist in our proteins that we haven't yet drugged through methods like artificial intelligence and machine learning. We're not there yet, okay? One example is shown here. So this is a protein that many of us are very familiar with in the scientific world. This is cereblon. This is a protein that Tom Daniel and his colleagues at Celgene pioneered IMiD drugs for, like lenalidomide, that are used to treat multiple myeloma today, transformative therapies. These drugs bind in a pocket shown here on the protein that's referred to as the IMiD pocket, okay? I would call those orthosteric drugs because they bind in the same pocket that the natural substrates that interact with cereblon bind, okay? There are hundreds, maybe 100 structures in the public database of small molecules bound to that pocket. Our activity-based proteomic work has discovered at least 2 other pockets on this protein that can bind permanent chemistry very far away from this IMiD pocket. We would call these allosteric pockets because they can regulate the protein's function in a way that's distinct from binding to this classical druggable orthosteric pocket. So then the interesting question is you ask, okay, what will an advanced, modern, artificial intelligence machine learning program predict as relates to where these ligands that bind these cryptic allosteric pockets bind on this protein? And without exception, it puts those ligands in the orthosteric pocket, okay? So this isn't maybe so surprising. These methods have been trained on data that says this is where drugs bind the protein, okay? So then when you give it a drug that binds the protein elsewhere, it says, no, 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 it should bind it here, because that's what I've been trained to learn, right? So I'm a runner, and so I oftentimes try to think about scientific concepts through the lens of athletics. And so if you train too hard in running, you burn out, right? So we call this in the lab orthosteric burnout, right? This is overtraining of artificial intelligence machine learning programs on structures that exist publicly that are basically 99% filled with small molecules that bind orthosteric pockets, right? And I'm showing this today because I am willing to wager anyone in this audience that we're going to spend 99% of our time in the next 50 years drugging allosteric pockets, right? So we need to solve this problem, right? This is how we drug proteins in the past. This is going to be how we drug proteins in the future. And I think there are ways to do this, and I'm happy to discuss them with you later if you'd like, but it's going to require new training sets for artificial intelligence to have an impact on these cryptic druggable pockets. Okay, so as far as where our lab is going, we really do believe that with innovations in chemistry from our collaborators like Phil and Dale and Jin and others, that we will be able to build a near complete map of cryptic allosteric druggable pockets throughout the proteome in disease-relevant proteins. One of the challenges then is to try to understand how drugs that bind those pockets actually affect the protein functions, right? That's not actually easy to predict today. Interestingly, the genetic maps of proteins are also getting equally rich, right? And so I envision a future where we will be able to predict the structures of almost all proteins. We'll know where our small molecules bind. We'll know where the genetic variants that cause a phenotype in humans when they're mutated also are located. And you can triangulate all that information to predict the druggability and the functionality of all these sorts of cryptic, shallow dynamic pockets that exist in our proteins and cells. And that day is coming, and that will be a very exciting day, right? Because we'll be able to say, oh, we found a ligand against this pocket, and now we know exactly what function it's going to have because we have genetic mutations that tell us that. And the ultimate goal is to convert that knowledge into precision mechanism-based medicines that can treat diseases with maximum efficiency, right? We want and we feel obligated to try to accelerate that process so when you and your loved ones go to a physician with the disease, it's not just a discussion of what caused that disease, but also what type of treatment can be used to restore that disease and restore you to health. Okay, just some final thoughts on Scripps. Whatever achievements our lab has made over the past 25 years, they are entirely due to this amazing institute. It was impossible for me to capture in a minute or two what's so special about Scripps, but I'll just share a couple thoughts. One of my favorite aspects of this institute is it's not a university, right? We don't have a football team. We don't have a humanities department. We don't have a band, right? We are singularly focused on biomedical research, trying to make fundamental discoveries, and when possible, convert those fundamental discoveries into medicines to benefit human health, right? And that singular purpose is so invigorating for us as scientists, right? From the president and CEO, Peter Schultz, from our board members like Tom, all the way down, right? We're all rowing in the same direction, right? And I love that about this institute. It creates a spirit of collaboration and collegiality that is, in my mind, unparalleled. I think the vast majority of students that work in our lab do so in a partnered way with other labs, Jin's lab, Jeff's lab, others across the institute, serving bridges between fields that would otherwise have historically been siloed. I want to call out Tom again, who created a collaboration fund to support these types of interactions at Scripps, right? A remarkably innovative approach where now investigators at Scripps can apply for funding, for seed funding, for projects, but the catch is the project has to be collaborative between more than one lab at Scripps, right? And that's the type of spirit of collegiality that exists at an institute like this, where everyone is focused on the same general objective. I'm also deeply appreciative that Scripps is so supportive of entrepreneurism amongst its faculty. Okay? We are almost all supported by NIH funding. Hopefully that continues in the future. Those are your tax dollars, right? And when your tax dollars fund work in our lab, and our lab makes fundamental discoveries that have a potential to impact human health, we are obligated, responsible for translating that information into those medicines, right? That's how we give back to you, hope that your investment in us can help you, your families, your loved ones enjoy healthy lives, right? And I think Scripps takes that obligation and responsibility more seriously than any institute or university I've ever been associated with. So with that, I will thank the incredible members of our lab. I learn more from these individuals than they do from me. They are tirelessly committed to their work. Collaborators, oops, sorry, that was a mistake. Collaborators like Phil, Jin, Michael Erb, and others here at Scripps. Collaborations with the local biotechnology companies that we've started. My original academic mentors, Dale and Richard, I would not be here without them. And my entrepreneur mentors, Tom and Rich and Paul Schimmel as well. Paul's a faculty member at Scripps who originally guided me through much of my interests in company formation and translating technologies from our labs into company formation. Finally, thank you for your attention, and I'd be happy to try to answer any questions you might have.
All right, thanks for the drink from the fire hose. That was great. So put your hands up, but let me just open up with a little bit of a softball. But you know, when you said we did our first screen, and then you get 747 hits.
Yeah.
You know, some number, hundreds and hundreds of hits.
Yeah.
Each one of those is a potential drug target. And so how do you actually go about triaging that list? Because you don't have.
That's right.
It's like you don't have 647 graduate students.
No.
You know, you can't possibly do this all.
Yeah, it's a great question.
So how do you triage your candidates?
So that's actually changed over time, right? So as I made reference to in the presentation, we struggled with that question tremendously in the early 2000s because we did not have the human biology information that we have today to prioritize proteins based on disease relevance. And we had to prioritize them based on superficial bits of information. This protein's higher expressed here than there, and almost all those bits of information invariably failed to allow us to pick proteins in a productive way as relates to disease relevance. But today, it's far easier, right? Because we can go through those lists of proteins that we have small molecule ligands for, as you mentioned, and we can align them with disease relevance based on the OMIM database that I mentioned earlier or the DepMap database that I mentioned earlier. And that can become a very rigorous way to prioritize a subset of those for a characterization. Now, of course, your view of what's interesting and my view of what's interesting may be totally different, and that's fine, right? So that's why we publish our data sets as a whole, and we do. I would say every couple months, you know, I get a phone call from someone who says, hey, I was digging through the supplemental table of your paper, and I noticed it looks like you have a ligand against this protein. And I'm like, yeah, we do, and I never really thought about studying that protein, and they're an expert on that protein, and that forms a collaboration, right? Where then they do most of the biology, but we provide them with their first chemical tool, right? So I call that anticipatory pharmacology.
Yeah, okay,
right?
good. Yeah.
You create the chemistry first, and then you anticipate, you know, going forward collaborations.
Anybody with their hand up? We got someone down here. We need a mic down here. Come on. While they're coming down, let me. We do have someone down here. Several.
We can repeat the question too if you want. So.
Yeah, where's the Jedis? There we go.
Hi, wonderful talk. So I actually wanted to follow up on one of the last remarks you mentioned on the productivity of Scripps with respect to entrepreneurial endeavors and the obligation of the taxpayer dollars translating into real social benefit. I look at Scripps as a real top decile producer of those results vis-a-vis other elite university institutions. What do you think other universities can learn from Scripps to improve their effectiveness in translating basic research into translational impact?
Right, okay. I'm probably going to end up regretting saying this, but the right way to say this. So, one of the things I've always loved about Scripps is they don't eschew conflict of interest. They embrace and manage it, right? And I think so many universities have gotten over their skis by trying to block and prevent conflicts of interest that they screw the taxpayer over, right? Why should it be the case that if someone uses your dollars to invent a technology, put that technology into a company, that lab should be firewalled from continuing to work with that company for conflict of interest reasons? That messes with your tax dollars, right? You want those groups to work together, right? To accelerate the transfer of the technology and the ultimate development of medicines. And of course, that creates conflicts. I'm highly conflicted, and we manage those conflicts, right? But that's to your betterment, right? And I think that's something that Scripps has always done incredibly well. And I think that's partly because we're a research institute, right? We don't have I hope there's no administrators here, but we don't have hundreds of administrators looming over our shoulders, right? Trying to micromanage what we do every day, right? And I think our administrative staff works for the betterment of biomedical research, right?
I think you missed one of the memos.
So I think that's a really important factor. I really do, yeah.
So let me follow up on the AI question, because lots of people might be aware of AlphaFold.
Yep.
And that's a tremendous program that can predict structures.
Yep.
But what you need is the small molecules bound. And they have forbidden people to do that with their software. So you can't do that.
There are public softwares that are getting...
So that's the question. And can you go ahead.
So yes, but that's not what we need. What we need is what folks like Professor Kern are doing, right?
Okay.
We need to integrate, well, you can yell at me if I'm misstating this, but I think what we need is to integrate artificial intelligence machine learning approaches, which are largely static analyses, memorization tools of known structures, with physics-based dynamics so that we can understand how proteins move to accommodate small molecule binding or in response to small molecule binding. That good? Okay, all right. So
good job.
To me, what's so amazing about these cryptic, non-orthosteric drug sites is they almost always involve motions, right? They're dynamic, the molecule binds, the protein moves, or the protein moves to allow the molecule bind, and that is where, static X-ray crystallography structures fall short.
Okay, I have a question here.
Okay, thank you very much. Okay, so what do you, are there efforts to combine activity-based profiling with peptide-based drugs, and what do you think are some challenges or opportunities that could open up?
Oh yeah, I mean, this is not exclusively the domain of small molecule, right? You can look at medium-sized peptide molecules, macrocyclic peptides with this type of approach. I know of several labs that are now embedding even permanent chemistries within biologics, right? To allow them to achieve a level through engineering, right? To allow them to achieve a level of irreversibility to their mechanism that wasn't possible before. And all those can be read out by the types of activity-based proteomic methods you described. So I think that actually is kind of a frontier field. Like, how can you leverage permanent chemistry and the types of technologies that can make you feel confident they're selective, right? In the context of biologics drugs, not just small molecule drugs. Yeah, so that's a great question.
So let me, I have a virtual question. I'm going to aggregate a couple of questions, but you obviously were very passionate about cancer targets.
Yeah.
And so in general, is this broadly applicable to other diseases?
Oh, yeah.
And what other, what's the promising other areas that your technology can assess?
Yeah, certainly the activity-based proteomic technology is agnostic, right? It can be applied to any biological system. The question of permanent chemistry has its strongest toehold in oncology when you move away from the sort of exceptional active site enzyme chemistries that I described before. But it's not exclusively the domain of cancer. There's a beautiful kinase inhibitor from Pfizer that operates by permanent chemistry to treat autoimmunity, right? And I think you'll see more of those drugs as they go forward. It's not surprising maybe that permanent chemistry in the pharmaceutical industry first branched out into cancer, because the safety expectations for cancer drugs are a little bit lower than more chronic disease treatments like metabolic disorders or autoimmunity, but you're beginning to see it branch out. And certainly the core technologies that I described today can be applied to any physiology or disease setting.
Yep, down here in the front, yep.
Great talk. I want to follow up on the first question from Jamie. And so once you have that target and the binder, I think the next critical step is really to identify what the role in biology is. And my question to you is, where do you see really new opportunities in terms of technologies coming up to make that translatability better? Because that was always the problem in cancer. Yeah.
You're talking about the translation of the pharmacology into the right kinds of treatment for a human, right? Yeah, I mean, that's still one of the major challenges that we face, right? And I do believe that there's great perspectives and reviews written on this by people who are even more religious about the topic than I am, that human genetic information is a tremendous filter for target selection, and that if you can have human genetic information that says when this gene is mutated, it will affect immunity, you're much more likely to be successful if you develop a drug against that protein in an autoimmune setting, right? And there's a lot of truth to that, but there's also incredible exceptional stories. I mean, as Tom and Celgene developed with the IMiD drugs, right? The cereblon protein that those bind to, it's actually not relevant to multiple myeloma at all. What it does is it creates a glue between that protein and other transcription factors that are drivers for multiple myeloma, right? There's no human genetics that would have said go after cereblon for multiple myeloma, right? And I think that's an interesting area for chemistry, right? Where you can create chemistry-first approaches for drug development. But I still think in the end, per your question, they almost always will dovetail back to a mechanism that can be understood as being relevant to the disease, right? And I think that's when I said at the very beginning of this talk, it's such an incredible time to be in biomedical research, is because we have these tools to understand at a molecular basis disease mechanisms that just were not available even 10 or 20 years ago, right? And it's just, you're gonna see so many more fundamental mechanism-based drugs entering clinic soon. I think they will have a higher probability of success. Yeah.
Okay, I think we've come to a pause. We'll take one more up in the back.
Hi, wonderful talk. I just wanted to ask really quickly about your slide on cereblon there at the end. I thought that was very interesting. Cereblon's used for many of the PROTACs we're seeing coming out today in one of the hottest fields. So using those allosteric sites for potential PROTACs, do you see that as a possible future, and do you see that helping us overcome some of the hurdles we're seeing with PROTACs today?
I would even generalize that question further, right? That a lot of the permanent ligandability sites that we've found, right, drug sites on E3 ligases in general, even beyond cereblon, are potentially attractive approaches to democratize, right, the targeted degradation modality approach beyond just the single E3 ligase. Now, there's a lot of criteria, as you probably know, because you sound like you're an expert in the field, for E3 ligases to satisfy to be useful for molecular glue or PROTAC-based targeted degradation, and cereblon checks a lot of those boxes. One that you may not be familiar with, which is really interesting, is the ligases that seem to work well for targeted degradation those proteins are parts of complexes that are highly efficient at degrading proteins, right? They have an incredible efficiency of assembly and degradation, and I think that's something we underestimated maybe initially at the outset, that there's a subset of these proteins that are horsepower drivers, and they're the ones that get discovered over and over again in screens for ligases that can support targeted degradation. But it may be a finite number, but it's not just cereblon. So I think those opportunities do exist, as you noted.
So I got one more really a good general question from virtual audience. And that, so you're a serial entrepreneur. You've had several companies. You've maintained a first cutting edge academic lab. And so you've obviously got a lot of scientific input in the companies. How do you balance the academic entrepreneurship in your life?
Yeah.
And keep it all,
Well,
compartmentalized but synergistic?
Well, I've never launched a company out of our lab that hasn't been in San Diego, and I never will. right? So that helps because they're local, right? And I want to support the local community too, right? I mean, you know, they often harbor members from Scripps that become employees there. You know, having said that, my roles at the companies are different than the role I play in the lab, right? And I've kind of learned that the hard way over 20 years, right? You kind of learn, you know, if an academic founder is driving the science at a company, they're in deep trouble, okay? They need to have their own leadership, right, who can make their own decisions, and then my job is to advise, consult, help where needed, but not intrude, right? And so does that take time? Yes, it does take time, but I would say I'm far more enriched by the interactions I have with the companies that I start than I enrich them, right? Because they're incredible people, and you know, and I mean, I think Jamie will agree with this. In academics, you know, we can launch projects with the idea of hitting a home run, right? And if we don't hit the home run and we hit a single or a double, we can get funding, we can publish papers, students graduate, all good, you know? In biotech, if you don't hit the home run, you kind of strike out, you know? And it's a very humbling and challenging experience to be involved with that, and I would say that I have so much respect for the leadership and the employees at biotechnology companies because, you know, they really don't have the ability to find an off-ramp the way that we do for partial success, right?
Well, I just learned something about you indirectly, which is you like baseball.
I do. I do.
Okay, let's wrap it up. So I want to thank you all for attending in person. You're welcome to join us for the reception. Thank you all to the virtual members of the audience, and we encourage you to come to the next one live, which is October 8th, and it's going to be Howard Hang, and he's going to talk about the microbiome, and everybody's got one, and everybody knows when it's messed up. And when Howard gives his talk, you will find me in the front row.